We go. Okay, thank you very much. And first of all, I want to thank the organizers for giving me the opportunity to present today. It's a real pleasure to be here and to have a chance to tell you a little bit about the science that we've been doing. I realize that the title I gave for my talk is a bit of a mouthful, so I have shortened it down a little bit just to say that my favorite channels aren't just channels. So that's what I'll try and convince you about today. So the largest family of mechanically activated ion channels, and my favorite family, one we've been working a lot on, is this family known as the OSCA or TMEM63 family of mechanically gated ion channels. So the OSCA side of this family is a group of proteins found in plants, and they're mechanically activated and play really important roles in things such as detecting drought or salt conditions around a plant. So when you have drought conditions, it creates an osmotic stress on the cell membrane that creates a tension that opens these channels, and the influx of calcium can stimulate the plant to do things such as close stomata and save its water. The plant channels, they're closely related to the channels that the TMEM16 channels that Will introduced earlier on. These function as dimers, and there's a lot of them. So even in the model plant Arabidopsis, there's 15 different versions of this. We don't really know why. Some of them are very low conductance, some of them are very high conductance. Now, mammals and humans have homologs of these. These are the TMEM63s. You can see here, these are interestingly monomers, not dimers. And they typically have very small conductances, but their physiological roles are still being explained. So they seem to have some kind of roles in hearing and nervous system development. That's due to the effect of mutations in these channels, but we don't really know exactly what they're doing. And so this was one of the questions that we were interested to find out. So we like to understand how proteins work at a very fundamental level, and looking at questions like what do the open states of these channels look like? Because when we started, we only had closed state structures. How do they respond to mechanical forces to open? And what's their physiological roles? Now, before I go any further, I want to highlight that this is really collaborative work from 3 different laboratories. So the structural biology I'll tell you about comes from Yizhao Zhang's lab in Shanghai. The functional studies are from Charles Cox's lab in Sydney. And then the computational work is done in our lab. And what I'll show you today is particularly from YC Lin. Okay, so we were previously part of a study that was trying to investigate the open state structure of these channels. And Yizhao's lab were able to do some beautiful cryo-EM looking at the OSCA, the plant channels, in liposomes. And as I said, there'd only previously been closed state structures of these proteins, but they were able to image the proteins in these liposomes. And depending on which way the protein oriented in the liposome, they found 2 different structures. So this inside-out conformation looked a lot like the previously determined closed state structures. But if the protein oriented the other way relative to the curvature of the liposome, they got a very different structure. And you can see top down, the dimer sort of, the middle part comes together and the outside part splays apart, and you get a very different conformation. And we wondered if this could really represent an open channel structure. But a very interesting aspect of their cryo-EM is if they looked at the density that they would typically assign to the membrane, so a low-pass filtered density, they could see the upper and lower leaflets in orange here of the membrane, but they saw this band running between the 2 leaflets. And they were wondering what this is. Is it really related to the membrane at all? And that is one thing that we could help to answer by running MD simulations, the type of thing that Will introduced. And if we took this new structure and put it in a simulation, we would see that lipids would in fact spontaneously move down this little groove in this protein and form this belt between the upper and lower leaflets. And interestingly, between these lipids and the protein, there was a water-filled pore. And if we apply an electric field, we'd see ions passing through this pore. So this was suggesting that this could be an open state, and a very interesting one in which lipids line the pore. And just to sort of prove this, if we take our simulation and get the density of the lipid head groups, we get this orange surface here that looks very much like the cryo-EM density. So that for us suggested this really was a band of lipids that moves along the surface of the protein. So this was our model of what this protein and what this channel looks like. So we have a protein here, our OSCA protein embedded in the cell membrane, but the channel is actually in a groove on the protein that runs between the protein and the surrounding lipids, and the lipids form this belt between the 2 leaflets. And to sort of give final proof that this was really a lipid-lined pore like this, we thought, well, this pore could in theory move cations and anions, but maybe we can influence that by changing the lipid composition. And because we were doing these studies in liposomes, we could change the lipid composition, for example, introducing negatively charged lipids, and thought, well, maybe that will change the ion selectivity. And that's exactly what we found in the electrophysiology. So this is just showing reversal potentials to measure the cation to anion conductance as we change the lipid composition. So with neutral lipids, we found it was about 2-to-1 cation selective. But as we introduced 20% negative lipids, we found this changed a lot, became much more cation selective. So for us, this was really sort of the final proof that we have this really interesting lipid-lined pore in this protein, in this open state of the protein. Okay, so that was the prior work, but what I want to focus on today is another question. So here we have this cool protein with this continuous belt of lipids that runs down this groove. And these proteins are actually structurally homologous to the TMEM63 proteins that we'll introduced. And some of these TMEM63 proteins are known to be calcium-activated lipid scramblases. What does that mean? That means that they're proteins that can move lipids from one leaflet of the membrane to the other. So we wondered, do the OSCA and TMEM63 proteins also facilitate lipid scrambling? So to answer that question, the first thing we did was to run MD simulations. So this is an example of coarse grain simulations that Will introduced earlier on, where we've taken this open structure of OSCA 1.2, placed it in a membrane, and you can see we get this belt of lipids that runs between the two leaflets. I'm just highlighting some of the lipids here for clarity. Some of them are colored. And you'll see that they move from one side to the other. They in fact go in both directions. They go up and down. And so this really looks to be a lipid scramblase, like a channel for lipids as well as ions. And we can quantify that. We can see that this open OSCA structure has a very high rate of lipid scrambling, higher in fact than the related TMEM63 protein, but it only happens in this open state where we get this groove opening on the outside of the protein. It doesn't happen in the prior closed states. Okay, so that's a simulation. Can we actually prove that this is happening experimentally? So to prove the lipid scrambling, we turned to an in vitro assay back in liposomes or GUVs. And the idea of this assay is to form liposomes with some fluorescently labeled lipids. So a small proportion of the lipids have this fluorescent NBD tag. Now, if you expose these liposomes to BSA, bovine serum albumin, that can bind to the fluorescent tag, the NBD, and it either quenches or removes the fluorescent lipids from the liposome. And so what you expect to see is after adding BSA, the fluorescence coming from the liposome will decrease. But because it can only access the outer leaflet, it would only decrease to about half the initial value. However, if you have a lipid scramblase in this vesicle, then lipids can move between the two leaflets. And so when we add the BSA, we expect the fluorescence to reduce much further because lipids can move from one side to the other. And this is what our results look like. So here's direct images of the liposomes, and you can see the fluorescence decreasing with time after adding the BSA. And this is showing it quantified. So the black line here is just an empty liposome. And you can see that the fluorescence decreases to a bit over half the initial value as the NBD labeled lipids are removed or quenched. But if we have the OSCA channels here, what we see, follow this red line, for example, is a much greater reduction, which is the signal of the lipid scrambling behavior. So here for 2 of these OSCAs, we see lipid scrambling. And as a control, we have a mechanically activated TREC channel that does not show any lipid scrambling. Okay, so I think at this point, we're pretty confident that the OSCA channels are lipid scramblases as well as ion channels. What about the mammalian TMEM63 analogs or homologs? Now, the challenge here is that we don't have any open structures of TMEM63. So Yixiao's lab tried very hard. They tried again in the liposomes. They tried in all kinds of conditions. They could never get a structure with this open groove. We just kept getting these closed state structures. So we thought about alternative ways. And of course, one way now is to turn to AlphaFold and see what AlphaFold predicts. So this is just an example of predicting 100 structures from AlphaFold. And we actually get quite a diversity in the structures. And how I plotted it here is similarity to the open OSCA structure I just showed you against on the x-axis and similarity to the closed TMEM63 structures. Each dot is one predicted structure, and the ones on the top left look very closed. They don't have this groove on the outside, and the ones on the bottom right do have this open groove. So it seemed that AlphaFold could actually predict possibly open structures of this protein. And if we zoom in on the more open structures here, we can see they sort of clustered into different groups. And so we took representatives from these different clusters and we ran the coarse grain simulations again. And you'll see in the movie down here, you will see lipids moving down the groove in this monomeric protein. And in fact, the number of lipids seems to depend on how open that groove is. So basically, as we move from left to right, we're going from closed structures to more and more open structures, and then the number or the rate of lipid scrambling goes up. So it looked like we were at least able to predict some possible open states of these channels, and maybe even some intermediates on the way with intermediate scrambling ability. Okay, so let's try and test these proteins experimentally as well. So this is the GUV assay again, with the fluorescently labeled lipids. You can see the empty liposome in black and the human TMEM63. So I should say we actually have three versions of this, TMEM63A and B. We're able to test both A and B, and you can see TMEM63A has this reduction in fluorescence indicative of lipid scrambling. TMEM63B also comes down, but not quite as much. So maybe it scrambles a bit less. And again, as control, we've got the well-known scramblase, TMEM16F, which is calcium activated, just to show what a scrambling signal should look like and what a sort of negative signal should look like as well. Okay, so it looks like TMEM63 is lipid scramblases as well. But interestingly, now that we had some predicted structures, we could start to examine what was going on structurally and mechanistically in these channels. And an interesting feature we found is that there seem to be different bottlenecks for the ion transport and the lipid transport. What do I mean by that? So the picture on the left is a snapshot taken from MD simulations, where you can see the protein on the left. The colored things here are this band of lipids that are moving between the leaflets. The blue here is just where water goes, and you can see the water-filled pore that goes between the lipids and the protein. And if we apply an electric field, we see ions moving through this pore. These pictures here are just showing, counting the number of ions that move through over time with an electric field. And you can see it's mostly sodium going through, a tiny bit of chloride. And the conductance is very low. That's consistent with the fact it's hard to measure single channel currents in these proteins. But the narrowest part of the pore comes from this W613 residue. So we thought, can we sort of get some more confidence in our structures by examining what happens if we mutate this? So if we mutate it to something a bit smaller, in this case we tried W613N, we see the current actually doubles, and it's mostly because chloride can now pass through this channel. But we could test that experimentally as well. So here's whole cell patch clamp experiments in hex cells with the wild type TMEM63A and the mutant. And again, we see this sort of doubling of the ion current. That suggests this really is a bottleneck for the ions. But interestingly, it's a different bottleneck for lipids. So here's a zoom in again of this groove where the ions and lipids go along here. This W at the bottom, this is the bottleneck for the ions, but it doesn't seem to get in the way of the lipids. And in fact, where the lipid head groups go, there's another residue here, W473. And so we looked at what happens if we mutate that one, either to make it more polar or smaller. And what you see is in our simulations, we get more lipid scrambling, indicative that it was a bottleneck. And interestingly, we don't see a change if we mutate the bottleneck for the ions. Now, could we test this experimentally? It was a bit difficult to do the mutations in our GUV assay. So we turned to a cell-based assay. This is one you might be familiar with. So we were trying to measure lipid scrambling by looking at phosphatidylserine or PS exposure on the cell. So it's often used when looking, signaling apoptotic cells where PS gets exposed to the outer leaflet through scramblases. You can use a fluorescent annexin conjugate that attaches to the PS lipids when they're on the outer leaflet. So the amount of lipid scrambling will relate to the amount of annexin that binds onto these cells. And I just want to show you that experimentally we saw something very similar to our computation. So in the wild type, we see annexin labeling indicative of lipid scrambling. But if we mutate W473 to a polar or a small residue, it goes up, and it does not change if we mutate the bottleneck for the ions. This was just a long way to say that there's different bottlenecks for the ions and lipids in this rather cool protein. Okay, a last little bit of fundamental information I want to tell you before asking why we have these proteins at all is just to note that cholesterol seems to inhibit the lipid scrambling in these proteins. So this is showing results again from the GUV assay, where we're looking at the change of fluorescence over time. Our initial GUVs just had the fluorescently tagged PC lipids, no cholesterol. If we add cholesterol, we no longer see lipid scrambling taking place at all. So lipid scrambling is inhibited by cholesterol. I can explain why, but I'll save it for questions later, but just given the interest of time. Okay, so we've shown that these proteins can move lipids as well as ions. Now they're well known to be mechanically gated ion channels. So the last thing we wanted to show is the lipid scrambling also mechanically activated in the same way, to really show that they're mechanically activated lipid scramblases. And so we did this both in a liposome and a cell-based context, but we needed a way to give a mechanical signal or create a mechanical tension in the cell membrane to open the channels. The way we've done it here in the liposome was to add alpha-cyclodextrin. So this is a ring-like molecule that can bind to phospholipids, and basically solubilize them, extracts them from the liposomes. And I'm not showing it here, but we can show that by asymmetrically removing the lipids, you create a tension in the bilayer. And when we do that, what we see again is a greater reduction in fluorescence in the presence of a scramblase like TMEM63A or the well-known TMEM16F. Sorry, but not in the well-known, only in the 16F with calcium. But what it's showing is that tension is activating the lipid scrambling. And we can do that the same in a cell. This is a hex cell containing TMEM63A. If we apply alpha-cyclodextrin, we see an increase in the annexin labeling, increase in scrambling, and we don't see that with other controls like mechanically gated piezo channel. And the final study of tension-induced scrambling I thought was a cool one is that these studies are done in hex cells, which are adherent to the adherent cells. So if we can grow them on a flexible cover slip, we can actually stretch the cover slip and create a tension in the cell membranes on the cells themselves. And so these results show what happens if we repeatedly stretch, like it's sort of a high frequency stretching. And the black and white images show the amount of annexin labeling, so the amount of PS exposure, an indicator of lipid scrambling. And you can see as we do this over time, we get more and more annexin labeling indicative of lipid scrambling. We don't see that in a control with mechanically gated piezo channel. It's something that seems to only happen with the mechanically activated TMEM63. So these really seem to be the first example of mechanically activated lipid scramblase. Okay, last question you might have is why on earth do cells have mechanically activated lipid scramblases at all? What's the point of having a protein like this? And there's many questions you might have, like what's the relationship between the ion currents? Which one is most important? But we wanted to think about what's the role of lipid scrambling. And one clue we got was when we were doing the liposome assays and adding the alpha-cyclodextrin, what we noticed is if you add a lot of alpha-cyclodextrin, basically you remove too many phospholipids and the liposome will burst. But if you have a scramblase in the liposome and you add alpha-cyclodextrin, they don't burst. Why is that? It's because the scramblase can redistribute the lipids between the leaflets and it will shrink as the lipids are moved, but it doesn't burst. And that suggested that they might play a role in helping cells respond to like toxic or harsh mechanical environments. So one way we could test that in a cellular context was to do the same thing in an actual cell. So we picked a cell line that had a high expression of TMEM63A in the plasma membrane. So in this case, it was a cancer cell, a human oligodendroma. Again, we give it a kind of toxic stimulus, in this case, a high dose of alpha-cyclodextrin, some toxic mechanical stimulus. In the absence of a scramblase, the cells lyse and die, but in the presence of a scramblase, they can stay alive. So this suggested to us that these proteins can help rescue cells in these harsh mechanical environments. We call it adding to mechano-resilience. The other role I haven't talked about here is when you're forming new lipid structures like organelles and so on, we think they're important there in redistributing the lipids in these newly forming structures. So that brings me to the end. So in summary, what I hope I've shown to you is that this family of proteins are dual ion channel and lipid scramblases. So the OSCA and TMEM63 move ions and lipids. And really interesting to understand what's the relative importance of each in different contexts. I think they're very cool proteins because they're different to a typical ion channel. We think of an ion channel as a pore surrounded by protein, but these are a groove in the protein, which is partly formed as well by lipids. So it's a lipid and protein lined pore. I think they're the first identified mechanically activated lipid scramblases, and we've suggested a role in mechano-resilience, helping cells survive toxic mechanical stresses. And with that, I just want to thank everyone that's been involved in the work. I won't go through them all again. And the last thing I want to note, I sneakily brought 2 posters. I put one on each side of my poster board. I have one on this topic if you want to ask questions, and another one to keep Will happy, a poster on predicting PIP2 binding sites as well. So very happy to show you both of those. And if I'm not there, please feel free to turn the board around and look at the other one. So thanks for your attention.
Thank you very much. Now this presentation of discussion. Is there a comment or question?
Hi Ben, great talk. How do you think it selects what type of lipid? Do you think each type of lipid permeate at the same rate across the bilayer? Because I think it's like a credit card swipe, right? As people always say, is that you have to swipe like that. But I'm always curious, is there anything interacting with the head group to make it specific or not?
Yeah, good question. So people typically think of scramblases as not very selective for lipid types, but you do have polar groups on the protein that interact with the head groups. We haven't looked extensively. We have tried it with things like PS, and all lipids seem to pass through it, but we haven't really quantified the rates of transport to properly quantify selectivity.
Yeah, because I was always thinking there must be a way to prevent the PS to be scrambled, you know what I mean? Because otherwise the cell will be very apoptotic. So therefore there must be some way to gate or lock it so that the PS doesn't just randomly.
Yeah, well, usually you just don't want to activate these proteins unless you have to, I guess.
Yeah.
So they are normally the way. So we have both the calcium activated and the mechanically activated cells, and normally activation of these, it is exposing PS, and that is a sign for apoptosis. So it is one of the steps in apoptosis. And so my answer to you is probably they're not selective against PS, yet they're just not opened too often. So in this case, if the cell's going to lyse because of a toxic mechanical stimuli, you can put up with maybe exposing some PS and hopefully locally.
And then survive for a while.
Yeah, and you might have a chance of surviving.
Yeah, thank you.
Very nice talk. And I'm wondering, because you show in the GUV experiment and like 20% of the cholesterol and inhibit the scramblase, and in physiologic membranes, and how much cholesterol on the plasma membrane, and do you think in different cell type might have the different role, and sometimes it open or shut down dysfunction, and also in intracellular membranes, it seems like intracellular membranes have much less closed role. So do you think in intracellular membrane probably they also function in different ways?
Yeah, lots of good questions. So mammalian plasma membrane, we do think of them having up to 30% cholesterol, so quite a high concentration. But yeah, can they operate differently with different cholesterol content? And I think you're right. I think that cholesterol may be something that helps to modulate these in a very physiologically relevant way. It's almost like a buffering system. The TMEM63B is particularly present in the intracellular organelles, and with lower cholesterol content, it may be more active. And that one we think maybe is more important in this organelle biosynthesis or biogenesis, I should say. There you don't have so much asymmetry and worrying about PS movement and things like that, and maybe you can deal with them being more active. But yes, don't know the answer to a lot of these questions. But yeah, very interesting how cholesterol might regulate them.
It's very nice though. For the TMEM16, for example, after studies, people said that just the head group of lipid just move through the pore, suggesting the clamshell model, something like that. So you show that all the tail part also looks like a path through the pores. How do you think about it?
No, so in this case, the head group is pointing towards the protein. So maybe I can show my hand. The protein's forming this kind of groove, and the head group is going partway into the groove, and the tail is staying embedded in the hydrophobic core of the bilayer the whole way. So maybe if I can make my arm, the hand is the head group, here is the tail, here is the groove. The lipid that starts like this, the head group interacts with the protein that flips around, but the tail is still embedded in the membrane core
Okay,
throughout.
so you think the same mechanism is
Yeah.
see here also?
Yep.
Okay, thank you.
Thank you. You mentioned the ion conductance was voltage dependent, and did you just see, because when you show the MD, there was a bidirectional movement of the lipid molecules inside or outside. So do you see the voltage dependent movement, and are they ever coupled to ion movement?
Yeah, good questions. We were interested in all of these. So we do see bidirectional movement of the lipids. Is it voltage dependent? We haven't really tested that. So it's a bit of yeah, I'll just say we haven't really tested that. We thought with things like PS that are negatively charged, maybe you could drive lipid movement with a voltage by moving the negatively charged lipids. Don't have the answer yet. Is it coupled to the ion movement? That was another one we were very interested in. And to our disappointment, they don't seem to be very coupled. So the lipid head groups don't go all the way and occlude the pore. They sit a little bit out and leave space for the ions. Yeah, and we wondered if they would move, the ions and lipids would move together, but definitely with Oscar, which has the wider pore, the lipids seem to be able to move independently of the ions. They could move past each other quite easily. I want to go back and look at the TMEM63, the groove's a bit narrow and there's less space, and double check this again. But so far it seems they can move independently. Yeah, but I'm curious whether something like a cation will interact with a PS lipid as it passes through or something like that.
It's very cool to see mechanically gated lipids movement, but I have questioned about Oscar and not really your study perhaps. So you mentioned there are many different varieties of Oscars in plants and their functions are not so clear, but some plants respond to mechanical force, and some plants, for example, leaves can close, right?
Yes.
Is it known whether Oscar is involved in that?
I'm trying to think of the right answer. I'm not a plant biologist, so I need to make sure I understand.
I'm just curious.
Yeah, I believe so, because they are the primary mechanosensors in plants. And it's definitely true that we know one form can detect an osmotic gradient in one way, and then one of the other homologs detects it in the opposite direction. So there's a little bit known about that difference, but why a single plant would have 15 different varieties, I don't really understand.
Right. Thank you very much.